When microplastics enter the lung of individuals who have COPD, it can cause lung damage, and actually it also changes the immune system response. And it is a big concern about the influence of microplastics on the health of such individuals. I'm Alexander Kell, and you are at the channel of No Coding Bioinformatics, and today we are going to analyze the data of gene expression on tissues from lung of different individuals and how microplastics is actually affecting this gene expression. And the question is if we can predict what is the effect of the microplastics on the gene expression system and on the immune system of those cells, and whether we can predict actually targets for the drugs which can actually tackle this problem. So what we are going to do, we are going to analyze the data from this entry of gene expression omnibus. This is data coming from this very recent paper published in the beginning of 2025. And here they were several patient samples from 4 asthma patients, from 4 patients with COPD, and 4 controls. These cells were treated with microplastics, one special type of microplastics, and they did gene expression studies using Illumina NovaSeq. And we are going to take this data, these counts, which were actually computed in the paper. We will copy this, and we are going to open our system. We are going to open Genome-Enhancer. So in Genome-Enhancer, let's start a new project. We'll call it COPD microplastic. So this analysis is done with Genome-Enhancer tool, which takes omics data and run a predefined pipeline which involve such databases like TransFoc and TransPos and human PSD, and it analyzes promoters of those genes which are differentially expressed, and then it reveals in the promoters of those genes, it reveals clusters of binding sites of transcription factors, and then by analyzing those transcription factor activity through the signal transduction pathways, it detects master regulators which actually control the expression of differential expression of those genes, and through analysis of these master regulators it predicts potential targets for drugs which can be used to inhibit or to change this response of those cells. Okay, so what we are going to do, we are going to upload this file with the counts in our system. So here it is. So we'll click import. We'll define that it's transcriptomics data and just click import. So now this file is actually imported into the system, and it's unpacked, and it detects which are the genes and what are the counts for each genes in each sample. So then we click next. And we see that this set of data is loaded here, so we can open this set of data. So you see this is 15,000 genes, where this gene expression measured in different samples. So what we do now, we drag and drop this set of gene here. We basically drag and drop this table in the system. Then we click next, and here we have 2 boxes for experiment condition and control. So experiment in this case is microplastic stimulation, and we'll select COPD samples. And control will be just COPD before stimulation. So we know that these samples 100 are actually coming from COPD patients, so we will drag and drop them, and correspondingly there are also 109, 10 and 13, 109, 10 and 13. So these are 4 COPD patient samples which were treated with microplastics. So then we click next. So next we select the disease, which is COPD. So here, pulmonary disease, chronic obstructive. And now we compare the microplastic treated COPD samples versus control, and we start the analysis. In a few hours we'll get the results, so well, let's see what we'll get. 52%. Okay, we see that the project just finished. So it generated a report. Let's have a look. Full story. All right, this is a full report of the analysis which was done. So the program analyzes the transcriptomics data and detects these transcription factors which are potentially involved in regulation of the genes, of expression of the genes upon treatment by microplastic. And when it analyzes master regulators, it detects these 2 master regulators as promising druggable targets in this context. So one can read this report, and you can see the initial data. And basically it detects 222 upregulated genes, which are listed here, and 184 downregulated genes. So among the upregulated genes, you see the keratins and mucin and other interesting proteins and genes, and it's very clearly you can see the expression patterns of our signal transduction genes is clearly distinguished between those samples which were treated by microplastic and which were not treated. So it's very clear signal. So here we see the basically those GO processes which were affected by this change of the gene expression. Among them we see the nucleotide metabolic processes, mitotic cell cycle, so very interesting, methylation and other processes related mainly to metabolism. So more specifically we can see which pathways were actually enriched by those genes which are changing expression. So cell-to-cell adhesion pathway, actually p53 regulatory pathway, number of chemokine activation pathways. And here you see actually comorbidities, like which biomarkers of different diseases were changing the expression. And many of those biomarkers related to the brain diseases like autism, epilepsy, but not only, so like child development diseases. As you can see, microplastic can be quite dangerous. All right, and there's a process which were down-regulated. But what is very interesting for us now is, in this context, is actually how the gene regulation is controlled by transcription factors. And this program, Genome-Enhancer, detects these transcription factors, these motifs for the transcription factors, such like HNF4, NF1, 1B, like AML, VDR. So very interesting factors which are in combination discriminate promoters of upregulated genes from the non-change genes. And here you see the factors which were involved in downregulation of the genes. So that's an intermediate step. And now when this program was doing upstream analysis from these factors, in order to understand what is the master regulators which can change activity of those transcription factors altogether. So these are, by the way, the list of potential transcription factors which are involved in upregulation of those genes, and you see those factors which I mentioned. And now here is a list of potential master regulators. And as you can see, these two molecules which we see in the title of the report, like the phosphatase DUSP8 and also kinase insert domain receptor, KDR, and that's another name for that factor, for that protein. These two molecules are at the top, and you can see actually the network, the diagram, how those molecules here, these master regulators, like VEGFR2, so that one which was detected, and also other master regulators, the pink molecules. Among them also you see here integrins, for instance, and here you see this DUSP8. And they are controlling through the intermediate molecules through single transduction network. They are controlling activity of these transcription factors like HNF4 and NF1 and RNX, VDR, which are in turn regulating activity of those genes, which I shown here at the bottom. So at the end, this report actually give a list of potential druggable master regulators. And again, you see this KDR, which is known to be quite druggable. So there are 70 known molecules in the market which are used to inhibit this protein in various diseases. And DUSP8 is not as well known as KDR as a druggable target, but according to our calculation, it's pretty promising target in this context. So you see here these two targets with those molecules, those drugs and active molecules which may be used to inhibit them. And you'll see this overall schema. Basically, then the program gives a report of potential drugs and which are actually used in COPD context. And it gives a priority of those drugs in the case of the COPD exacerbations which are triggered by the treatment or by inhalation of microplastic. Here you see the repurposing drugs, so potential drugs which can be studied in this context, and among them you see some of the anti-cancer drugs which are targeting those targets which was observed here in this analysis. And not only, there are some of them actually known drugs for arthritis or cystic fibrosis. So there's very interesting potential repurposing drugs which can be used for further studies. We can also see here some molecules like uracil, which can be predicted here as a potentially, targeting those targets which were found in this analysis. So basically, it's produced a very interesting hypothesis about the molecular mechanism of the changes of gene expression upon actually treatment by microplastic of the lung cells from COPD patients, and it gives some suggestions for the molecular mechanisms for master regulators and actually for drugs and active compounds which can be studied for as potential drugs in that context. Thanks a lot for your attention and stay in touch. Goodbye.